Hi all. So welcome back for another sample of NEET MCQs discussion for the upcoming unit. And this session will be completely based on molecular biology and genetics with your faculty on behalf of ADRPLEXUS, Dr. A. Balasubramanian. So let us not waste time and get to our MCQs. This is what is our MCQ sessions for discussion today. We'll have 10 MCQs. Okay, all the 10 will be solely from molecular biology and genetics. And before, as regularly, before we go into our discussion part, we will discuss some basic cruxes about how to handle molecular biology genetics. Okay, so molecular biology genetics as such. The central dogma of molecular biology, the entire questions, everything will be focused only on the central dogma of molecular biology. There are only three things which are to be remembered. Okay, DNA. This DNA will give rise to more DNA. This process is called as replication. Okay, so MCQs will be focused on replication and of course sometimes based on structure of DNA, structure and organization. Okay, so this will be one. This will be 2. Okay, so structure and organization of DNA, the basics are always there. Okay, nucleotide chemistry and metabolism. So since that is coming in chemistry, you have learned everything previously. I'm not going into that, but still the MCQs will be there. We'll discuss that also. So structure and organization of DNA. DNA multiplies to give more DNA, that is called as DNA replication, and then DNA is converted into mRNA. This is transcription. So area number 3. All right, and then mRNA finally gives rise to protein. Okay, so area number 4, crux area number 4. Okay, and molecular biology and genetic techniques. This is area number 5. This is area number 5. So these are the 5 areas in which predominantly you will get MCQs along with the nucleotide, very rarely nucleotide structure, chemistry and all those things. So our entire discussion should span, if you start attending our molecular biology classes, then your discussion will be oriented along, aligned along these topics so that you'll have a holistic idea about what is happening right from the genetic material which is stored in your chromosome as genes, DNA, and from DNA where it is ending up after being translated into a protein, and the protein is targeted and the protein is sorted and everything is like that, along with the latest advancements in the molecular biology technical fields. Okay, so this area is very attempting initially. Okay, all the premier institutions are focusing with more concepts and more questions towards the latest advancements, latest genetic technologies, latest genetic instrumentations, and everything in their questions. Okay, traditional NEET MCQs along the first 4 lines of topics, whereas the initial exams will focus more on techniques per se. Okay, so with this backdrop, let us go ahead and start our questions. Okay, all right, a 46-year-old male, I'll underline the pointers. Last time itself I told you what are the pointers, what are the distractors, and what are the stem of the MCQ. So the pointer is 46-year-old male presents to the emergency department with severe pain in the right big toe. Okay, right big toe. So most of you will get the answer by this time. He gives history, he has consumed excess alcohol. So alcohol is there previous night. On examination, the right big toe was swollen, warm, red and tender. The synovial fluid was obtained and viewed under polarizing microscope. Which of the following statement are wrong in this patient? So they're asking us to find out the wrong statement. All right. So the patient is suffering from gout. Okay, first option. Deficiency of HGPRTase can lead to his condition. Second option. Calcium pyrophosphate dihydrate crystals are seen is third option. Patients may have acid levels. So it's a pretty straightforward question, but only when you select the options, you have to be little careful. So let us take one option by another option. So 46-year-old male, pain in the right big toe, excess alcohol, all points to only one thing. It's nothing but gout. So the first option is correct. Okay, so we all know that how gout presents, how the gouty arthritis presents. It presents mostly in the great big toe, and that is because of accumulation of crystals, certain crystals. What are those crystals? The crystals that are accumulated is called as monosodium urate, or MSU. Now these crystals have particular, see what will happen, I will tell you. During the night, okay, see most of the times the classic picture is during the night. During the night means during the night the body pH will slightly shift to the acidic side, okay, slightly shifted to the acidic side. So the body pH is slightly towards the acidic side. When the pH is acidic, there is always tendency of the uric acid, many things, to precipitate as crystals. So basically the substances tend to precipitate as stones. The substances tend to precipitate as solids during the acidic pH, acidic pH environment. This is the basic reason why in renal stones, in order to end this acidity, we give one syrup called as citralca syrup. Why? Because citralca makes the urine alkaline. In alkaline pH, the stones don't tend to precipitate. So that is the reason. So always remember, acidic pH tends to precipitation. So the solid or strong is more favorable during the night. Okay. Second thing, it happen when you take alcohol is this enzyme called as alcohol dehydrogenase. It will be say for example ethanol. If he is taking ethanol, then this ethanol will be metabolized to aldehyde. What aldehyde? Acetaldehyde, by an enzyme called as alcohol dehydrogenase. Okay, and then this acetaldehyde will be metabolized by another enzyme called as aldehyde dehydrogenase to acetic acid. The problem is not with the acetic acid when it comes to gout. What is the problem here is because of this enzymes, aldehyde dehydrogenase, alcohol dehydrogenase, and aldehyde dehydrogenase, the NADH to NAD ratio increases. Okay, so both these enzymes take up and convert the NAD into NADH. There will be more NADH will be produced. But there is a problem here. When the NAD ratio is more increased, it goes and converts lactate. So it's an enzyme. Which enzyme? Lactate dehydrogenase. That's an enzyme here. So this enzyme uses up NADH and converts into NAD. So more NADH is available. No, this will come here. It will allow go and tell the pyruvate, get yourself converted into lactate. Now the problem is this lactate is a competitive inhibitor of uric acid. Then what will happen? This lactate will go and tell to the kidneys, see, you must utilize me, lactate or lactic acid, both are same. You must utilize me and you should not excrete that uric acid for low. So this will turn more excretion of lactate, thereby lesser excretion of uric acid. That is the reason why uric acid tends to accumulate in the body. So there is more tendency for precipitation. There is more uric acid. Where now the problem is where that uric acid is going to accumulate. It is going to the joints and it is going to accumulate there, precipitated. That crystals are going to hurt the synovial membrane. That crystals are going to hurt the joint. So that is the reason why you are developing pain. So monosodium urate crystals are the main reason for causing the pain. Now, how this monosodium urate crystals can be identified? This monosodium urate crystals can be identified by certain properties. Number one, this can be viewed only by polarizing microscope. Sorry. Polarizing microscope. What is polarizing microscope? A polarizing microscope is a microscope which will produce light that will be reflected by the structures viewed under that. Okay, so this polarizing microscope will produce a light that will be making the crystals to be visualized as yellowish green needle shaped crystals. Gout crystals will be viewed as yellowish green needle shaped crystals, and then there is a particular characteristic for that, that is called as negatively birefringent. What is meant by that is, see, it's too technical, but let me tell. The axis of the light passing and the crystals visualizing is, to the opposite side. See here. See, the crystals are visualized to this side, right? To this side. Okay, but the light will be passing from this side to this side. So this is called as negatively birefringent crystals. These are the crystals, yellowish green crystals. Okay, so this is called as negatively birefringent. So monosodium urate crystals, yellowish green, viewed under polarized microscope, negatively birefringent crystals. Now, this calcium pyrophosphate dihydrate crystals, where are they seen? They are seen in an entity called as pseudo gout. It is not gout. In gout, there is uric acid crystals, whereas in calcium diphosphate crystals are pseudo gout. So this is the wrong statement. Option is C. Come to the last option. Patients may have normal serum uric acid. Of course, they will most of the time have normal uric acid levels. That is because most of the uric acid is going towards the joint and is getting precipitated there, right? So in the blood, if you take uric acid levels, it will be normal. But most of the students are under the opinion that uric acid levels will be high in gout. Uric acid levels are high. All of them have gone to the joints. They are precipitating over there. In the blood, 50% of the times, almost half of the times, we see normal uric acid levels in gout. So this solves the question. This answer is calcium diphosphate crystals is seen in pseudo gout. They are not seen in the gout, right? Question number 2. I told you this. So the first question is from purine catabolism, right? Second question is from the purine structure. Okay, so this is purine structure. One more thing I have forgotten to explain, sorry for that. Deficiency of HGPRTase can lead to this condition. Yes, it can lead. That is because deficiency of hydrogen HGPRTase means what? That is salvage pathway, right? So where is HGPRTase? They involved. HGPRTase is involved in HGPRTase. What is the meaning? HGPRTase. Hypoxanthine guanine, okay, ribonucleotide transferase, okay, phosphoribonucleotide transferase, phosphoribosyl transferase, okay, phosphoribosyl transferase, which means hypoxanthine is converted to inosine monophosphate, guanine is converted into guanosine monophosphate by this enzyme. Okay, now if this then called as xanthine oxidase. You see the purine pathway, it will be okay. Xanthine oxidase will break down these products into uric acid. So more deficiency of HGPRTase can lead to increase in the uric acid by enhancing the uric acid or purine catabolism, thereby leading to formation of more uric acid. So that's option number 2. All right. So here, let us come to this structure. This question is one of the most repeated questions. This is a structure of a particular nucleotide which is given, or nitrogenous base, nucleotide nitrogenous base which is given. And let us see what can be derived from this picture. First thing foremost, you must remember, if the name is long, then the structure is short, simple. If the name is small, the structure is big. Okay, purine. The name is simple, so structure is big. 2 rings, structure, big structure. Okay. Pyrimidine. Name is big, so structure is small structure. Okay, so by knowing this concept, you look at this question, you see that 2 rings are there. So this is kind of some purine. This is kind of some purine. Total, it's a 2 rings are there. Okay, so what are the purines? The mnemonic is AG. Adenine and guanine. What is the pyrimidines? Pyrimidines mnemonic is CUT. Cytosine, uracil, thymine. Cytosine, uracil, and thymine. Okay. In this, thymine exists only in DNA. Uracil exists only in RNA. That is another MC you be knowing. Okay, I'm not going into that and all. Let us see the structure. Okay, and let us assume what is happening here. Okay. C4, C5, N7 are derived from glycine. Option number 1. C6 is derived from N5N10 methylene tetrahydrofolate. N1 is derived from aspartate. N3 and N9 are derived from amide nitrogen of glutamate. See, nobody knows. Okay, unless you know about the structure. There are very rarely structural questions are being asked. One other question which can be asked is cholesterol. Cholesterol structure, you have to know the intricate details. Some 5 points are there for cholesterol structure. Other than this purine structure is important. Okay, so this purine structure, you have to again remember 5. What are those 5 points? If you remember, it is very easy to understand. So these are the 5 points which you are going to remember. See, C4, C5, N7, all of them come from glycine. Point number 1. Okay. Point number 2. The nitrogens in position 3 and position 9. The nitrogens in position 3 and position 9. They both come from glutamine. They both come from glutamine. Okay. Then the carbons. Look at the carbons. The carbons at position C8. Okay. And C2. Sorry. You can write like this. C2 and C8. C2 and C8 come from folic acid. Folic acid are their derivatives. Tetramethynyl tetrahydrofolate or format tetrahydrofolate, all these things. Whereas the carbon at C6 comes from carbon dioxide. Some people say that bicarbonate or carbon dioxide. So, okay, so carbon dioxide. Okay, that leaves us with only one nitrogen. Okay, 4 nitrogens are there. Okay, so which is the only one nitrogen? Aspartate. So nitrogen 1 comes from aspartate. You have to remember this. Okay, you have to remember this. C4, C5 and N7 go together. They are forming the glycine. Okay, whereas C6 comes from carbon dioxide. Okay, C6 comes from carbon dioxide. C2 and C8 come from folic acid. They are folic acid derivatives. C6 and C7. Okay, sorry, C2 and C6. Okay, remaining nitrogens, N1 comes from aspartate, N3 and N9 comes from glutamine. So we need glycine, glutamine, carbon dioxide, folic acid, and aspartate. These are the starting materials for, these are the substrates that are necessary for purine synthesis. All right. So now let us come to the question. C4, C5, N7 derived from glycine. Correct answer. C6 is derived from N5, N10 methylene tetrahydrofolate. No, that is wrong. C6 is derived from respiratory carbon dioxide. Carbon dioxide is the source for C6. N1 is derived from aspartate, correct. N3 and N9 are from glutamine. That is also correct. So if you know this concept, then you will understand and answer this question. All right, fine. True about nucleosome is that, okay, let us, I will first go ahead and I will explain you one thing. Give me a minute. We will add an empty slide because we need an empty slide. Okay. Okay. See here. Understand this concept very nicely. DNA is a negatively charged protein. It's a not protein, negatively charged substance. Okay. It will always go ahead and interact with a positively charged substance. Okay. What is this positively charged substance? That positively charged substance is histone. Another category. DNA is mildly acidic. Histone is mildly basic. Okay, sir, that's all okay. Why do we need this DNA and histones to be together? That is because of a word called as organization of DNA. Why do we need to organize DNA? Because if there is one finding, the Human Genomics Project, when they started decoding the human genome, they came up with a full sequence, genomic sequence of human body. Okay, so they said that if you start arranging the bases one by one, side by side, parallelly, these bases, if they are arranged like that, a single man's genome can stretch from Earth to Moon. See, that is the length of DNA which your body is carrying. Okay, so this is compactly packed inside the cells. There is no space for that much to exist freely. So it has to be packed. It has to be condensed. So this condensing packing is possible only because of this organization of DNA. So how the basics of organization of DNA work is, this DNA interacts with the histone and it is looking like an one by one they are linked together to form a structure called as nucleosome. So you must understand what makes up the nucleosome. Before going to that, histone molecules are divided into, there are totally 5 types of histones: H1, H2A, H2B, H3, H4. These 4 together as group H2, 2A, 2B, H3, H4. Let us assume like this. These, each of them is a dimer. Dimer means 2 subunits. So totally 8 subunits are there. 4 into 2, 8. Okay. This is H2A. I am drawing another one round. Like you just assume this is another dimer of dimer. Okay. H2B, H3, H4. Okay. Dimers. Okay. Now, this is a histone molecule, and then our DNA, because of its affinity, comes and voids around. It just encircles this histone. This unit, which is a combination of DNA encircling the histones, is called as a nucleosome. This is called as a nucleosome. Now, this nucleosome, one unit is attached to another nucleosome by a linker. By a linker molecule. This linker molecule is called as H1 histone. So H1 histone is basically a linker molecule which connects one nucleosome to another nucleosome. Now you'll ask, okay, sir, that is all fine. Now how is this nucleosomes connected to each other? All right, everything is fine. So these nucleosomes, they are all connected together. They form something called as chromatin fiber. Then this chromatin fiber will lead to something called as chromatin fibril. Then this chromatin fibril will lead to something called as chromatin. As you know, chromatin structure, the chromatin structure. Okay, there are many intermediate structures. Okay, chromatin scaffold, chromatin fiber, chromatin fibrils, finally chromatin. All the chromatins together, put together, they form the chromosomes. So they are located in the chromosomes. This is how the genetic material is tightly packed with each other, condensed together to form layer by layer of arrangements, and finally ending up with chromosomes located in the chromosomes. Now let us go back and see the question. Okay, see here, this green color is H1 histone. Okay, the red color tape-like thing is the core DNA, and then there are 4 histones, 2A, 2B, and all these things. So everything is mentioned in the picture. What is it? True about nucleosome is that it is true. They're asking true statement. It uses only one type of histone protein. No. Each complex is separated from each other by non-histone proteins. No. It contains regular repeating structure of DNA and histone protein. It reflects small nucleus. No, the structure is very big. It reflects a big nucleus. So the only correct answer here is it regular repeating structure of DNA and histone protein. Yes, this is the correct answer here. Okay, fine. Identify the technique depicted below. Southern blot, northern blot, eastern blot, western blot. Nothing against you, but you have to see the picture. Most of you know what is this thing. Okay, see, first what we'll do is, I will tell you, in labs, we use, I particularly use something called as western blot. Let me tell my experience. Western blot. Okay, there are 2 procedures for which we use western blot widely. I'm speaking about commercial laboratories where we work, not in the research laboratories. Research laboratories are totally different here. Altogether, the game is commercial laboratories plays. They use western blot to confirm the PCR done for, confirm the ELISA done for HIV. That's one thing, point number 1. And then point number 2 is we evaluate antibodies. Western blot, we do Western blot to evaluate antibodies for diseases like SLE. You all know, no? Anti-lupus antibodies, anti-Ro, anti-La, anti-ISIS, anti-nucleus antibodies. Okay, all these things we do. We take this Western blot technique to evaluate that antibodies. So the answer I have given here. But now you look at the picture. So we take antigen samples. Where is the antigen? The antigen samples, or whatever it is, the proteins or whatever, the blood, the substance which you need to evaluate. Those are the antigen samples. We run some electrophores. So the first step is electrophores. We separate them. Basically what we do, we separate them using electrophores. Okay. Then all the proteins, once they are separated, we transfer them to something called as a nitrocellulose membrane sheet. Okay, nitrocellulose membrane or nitrocellulose sheet. There we add labeled antibodies. So these are antibodies which are labeled. Why we are labeling the antibodies? There are other many techniques for labeling the antibodies. You can label them with fluorescent markers. You can label them with enzymes. You can label them with color substances. All these things are labels. Why we attach labels is when the antibody binds to the antigen, these labels will start giving an indication whether they will change color, whether they will react or convert the substrate to the product. All these things will happen. So that's why we put some labeled antibodies there. So this is called as immunostaining. This is called as immunostaining. So what will happen is the antigen is there, then we add some antibodies, labeled antibodies, then the secondary antibody, the antibody which you need to evaluate, will come and bind to this labeled antibody. It will come and bind to the primary antibody. Okay, so this antibody is basically what we are looking at. So I told you, no, anti-Rho, anti-La, everything we are looking at. So this is the antibody which we are targeting. Okay, so we take an antigen, we put some antibody, then this antibody is designed something like it will capture the secondary antibodies. Okay, so now this is all subjected to radiography. This is all subjected to radiography, and then we finally see the bands. So we start with the protein, we find out the antibodies to that particular protein. In this case, this kits are designed to give the proteins, and then the labeled antibodies as reagents will be given to us. We open the pack, all this will be found. Then we take the blood sample and we start doing the procedures. Everything will visualized. Probably if it's in a class, I will show you some known patterns of ASLA antibodies or lupus antibodies, which we have evaluated in our lab. Okay, so everything is clear now. So this is very clear. This picture speaks about antibody detection using a blotting technique. So this is a blotting technique. You run electrophoresis, separate the material, then you transfer it to a nitrocellulose membrane, then you add antibodies to that, then you find the secondary antibodies which are targeted against this primary antibodies. In the case of other blots, as per the materials we will design the kits, and as per the process we will identify the substance to be identified. So this is antigen antibody. So antigen antibody detection is done by only one blot, which is called as Western blot. Okay, proteins. Some people say that proteins, antigens are proteins. Okay, the answer is Western blot here. Okay, what are the techniques to identify the other substances? Okay, so there's a scientist who was called as Robert Southern. This is a guy who developed a blotting technique to identify DNA. So in order to place opposite as that, or somebody called that as a laboratory jargon. Okay, jargon is a crude name. Okay, they named the RNA blot as Northern blot, just because the DNA blot was named as after him, Robert Southern blot. They named the RNA as Northern blot. And Western blot, most of you would have known by this time, it is against antigen antibody or protein antibody reactions. What about the Eastern blot? Okay, Eastern blot is something to analyze post-translational modifications in proteins. After translation, after the proteins are synthesized, what are the modifications now? That is called as Eastern blot. Once in AIMS, they are asking one question: what is meant by Southwestern blot? They'll ask everything. Southern blot is for DNA, Western blot is for protein. So this Southwestern blot identifies the proteins which binds to the DNA, which are called as motifs. So there are three motifs: zinc finger motif, leucine zipper, and helix and helix motifs. So these are not three motifs. There are many motifs. These are the primary motifs. So this is all about blotting techniques. All right. Good. So molecular biology technique number 2, identify the correct order of steps in polymerase chain reaction. So everything is now simple. So everything is now very simple. They have given you the PCR. Okay, they are just asking you the steps. If you know the concept, it's easy. Okay, so always remember in PCR, I'll tell you a story otherwise. There is in a park known as Yellowstone Park in USA, Yellowstone National Park. There's a guy called as Carey Mullis. Now, Carey Mullis decides to take a walk in the Yellowstone National Park. He sees the Yellowstone National Park is filled with volcanoes. Okay, so the base temperature itself is hot. Okay, there are certain, he gets a vague feeling, because when you read about enzymes, you will always remember that the enzymes get denatured when they are beyond their enzymes survive in 50 to 60 or 55 to 65, some textbooks say in this range only. However, how these fellows who are sitting on the base of a volcano with so much heat, certain plants are there, this plant is surviving. He was very puzzled. So what he does is he takes the plants, comes and takes and isolates their DNA. Now this plant which he brought with him is called as Thermophilus aquaticus, okay, or TAC. And the enzyme which he identified or which he isolated from them was named as TAC polymerase. Okay, TAC polymerase. That's it. He knew that it was a heat stable enzyme. So this enzyme is not going to get coupled by the heat. That it is already having an idea. Now this was the basis behind the developing of the molecular biology technique, which is called as PCR. Okay, so this is the basic story. Okay, now in PGI Chandigarh exams, they keep asking certain questions. One of the statement there, it's never about questions. No, in PGI, it's always about the answers, options. So each option is a question there. Okay, so one of the statements caught my attention, which is that always DNA anneals or develops a sticky property when you cool it, when at a lower temperatures. Okay, this principle was exploited in PCR. This particular characteristic of DNA was deployed to develop PCR technique. Okay, we will now see. Okay, I will just keep a overall bird's eye view here. There is a double stranded DNA. For those who don't know why PCR is done, let me tell you. See, PCR is coming under a molecular biology technique called as nucleic acid amplification. So even if one single molecule, PCR can be done for anything. Okay, we'll consider DNA here. Okay, even if one single piece of DNA is there, this one will multiply into 2, 2 will become 4, 4 will become 8. So they will run a particular type of cycles, particular number of cycles, 42 to 45 cycles or something. By the end of those cycles, a count will be obtained. They are going to look at that count and they are going to say whether the nucleic acid copy existed or not. See, it's being run routinely for coronavirus, right? RNA virus. So they are taking RNA PCR. They are running it, coronavirus PCR. So since because you're speaking about coronavirus, I will tell you there is a value called as CT value. If you look at the PCR reports, CT values na cycle threshold values. There's a particular amount to be quantified as you are quantifying as positive. There particular amount you are counting as positive. Okay. What the basically ICMR, they keep on revising the criteria. Okay. So supposedly in very less number of cycles itself, you are reaching that threshold value. Okay, you are reaching that, you are become positive. If you reach the threshold value, you are become positive. Okay, now the only question remains whether you are reaching it in lesser number of cycles or more number of cycles. So there are 3 persons, A, B, and C. One person is hitting the threshold value as 17. Another person is hitting at 21. The other person is hitting at 35. Okay, total number of cycles run is, let us assume 42 to 45. Now, this person is hitting the threshold value at very less number of cycles, which means starting itself, he had more material so that he became the positive at a very less cycles. This person is hitting the threshold value at very less. So the quantification of the material is very less in the start. Because of the amplification, he has reached the threshold at 35 number of cycles. Okay, now this can be extrapolated crudely to know about this person will have less viral load, and viral load is directly proportional to the transmission. Okay, transmission rate. They're not worried about the sickness. Sickness is totally about the immune system of that patient. Okay, so patients who are having more viral load have recovered. Patients who are having less viral load have expired. That all we are not speaking. The tendency for transmission is more when you have more viral load. The persons who are with him in the family has more tendency to get the virus if you are coexisting in the same habitat. All right, so this is the basic of threshold value. This is just about an information I thought I will give you. Okay, so first there is a double stranded DNA. The first step is unwinding the DNA into 2. Okay, each strand is divided into 2. This is being done at a temperature of 95 degrees centigrade. is being done at a temperature of 95 degrees centigrade. Now, once the strands are gone, what you're going to do is, for starting the replication, if you know about DNA replication, you need a small piece of starting material, which is called as primer. Okay, now this primer is put along with the nucleotides. Okay, primer. So the PCR reagent will be like this: primer, nucleotides, all these things necessary for, and then that enzyme, DNA polymerase enzyme. Okay, all these things are given in the PCR packet. Okay, so all these three you put together. You put the primer, you put the nucleotides, you put the enzyme. So the nucleotides will start complementary synthesis in this direction. Okay, to this direction. All the nucleotides also we have provided. But this starting material, primer, has to go and anneal with the DNA. How it will anneal with the DNA? It will anneal because when the cooling happens, I told you, no, when you cool down the temperature, the annealing will happen. Okay, so once the annealing happens, you put the nucleotides, you raise the temperature to 72 degrees centigrade. What will happen is the synthesis will happen. So if there is one DNA, now it has become 1 plus 1, 2 DNA. Next cycle it will become 4. Next cycle it will become 8, like that. Okay, so the process here is, first is separating the strands, denaturation. Second is annealing. Third is extension of the strand. So which is the option here? Just look at it: denaturation, annealing, extension, extension, denaturation, annealing, annealing, extension, denaturation, denaturation, extension, annealing, denaturation, annealing, extension. So option A is the correct sequence of reactions happening in PCR. Okay, the other molecular biology techniques which are involved in nucleic acid amplification techniques are ligase chain reaction and NASBA, nucleic acid sequence-based analysis. Then there is another branched chain DNA estimation also, but don't worry about this. PCR is the core crux technique which you have to lay your eyes upon. There are many types of PCR. Please read it for yourself. All right. Now, question number 6. Identify the protein synthesis inhibitor that prevent the attachment of amino-acyl tRNA to the A site of ribosomes. This is very, tricky. Okay, so I have to explain you this, right? 1 second. So we'll use this sheet, rough sheet. Okay, so translation is a process which I told you, it is divided into 3. It converts the mRNA into protein, that you all know. Now, the subdivisions are initiation, elongation, and termination. Now again, it's a huge topic. Okay, when I teach translation, I'll take more than 45 minutes to teach translation. But then simply put, to think simple, initiation, see, basically you must understand the ribosome structure. What is meant by ribosome? A ribosome is nothing but it's a combination of rRNA plus peptides. Okay, so and this ribosome is divided in prokaryotes and eukaryotes into 2. In prokaryotes, the subunits are 30 and 50. In eukaryotes, the subunits are 40 and 60. Okay, however, simple mathematics does not work. 30 plus 50 is equal to 80 in normal life. In molecular biology, it is 70s. 40 plus 60 is 100 in normal life. In molecular biology, it is 80s. Yes, what is meant by yes? Yes is meant by Swedberg units. When they centrifuged, differential centrifugation of the organelles, the ribosome started separating into subunits, and these subunits, when they put together, but then the molecular weight was not additional, exact addition, it tends to reduce a bit. Okay, this is Swedberg is the person who found out this. Okay, he told the whole prokaryotic ribosome weighs 70s, but if you separate them into subunits, one is weighing the 30s, the other one is weighing the 50s. Okay, so here we have it's like this, 30s and 50s. Okay, and then there are a lot of initiation factors which come together. Okay, but the basis here is there are 3 sites here. Okay, the mRNA threads through the 30s unit of the prokaryote. I'm not speaking about eukaryotes. I'm speaking only about prokaryotes. Since because we are dealing with the bacteria, antibiotics and bacteria question, I'm speaking only about eukaryote. Eukaryote also principle is same. Only the factors involved will be different. Okay. So the mRNA, as I progress, I will keep telling about the MCQs also. The mRNA threads through the 30S subunit of the ribosome. The place at which it enters is called as the Shine-Dalgarno sequence. Shine-Dalgarno sequence. The corresponding point in eukaryotes will be called as Cossack consensus. Don't worry about that. At least you know that the mRNA enters through the 30S subunit of ribosome in prokaryotes as Shine-Dalgarno. Now, there are three sites. You told sir 3 sites. One is A site, another one is P site, another one is E site. Okay. So what will happen is, in order for translation to occur, we need two materials. Not two materials. What is it? One is a ribosome. The other one is called as charged tRNA. Not verum tRNA. Not only tRNA. Charged tRNA. Charge means what? Like mobile phone, you put charger, that electricity is going and charge is there. For tRNA, charge you put means amino acid is loaded. Amino acid is loaded to the tRNA. Okay. So that corresponding amino acid which will be loaded will be, if you know the tRNA structure, there is something called as an anticodon arm. The tRNA structure, this is the anticodon arm. Okay. Anticodon arm. Okay. So the amino acid will be loaded here. It will be corresponding to the anticodon arm. Okay. Why? It will not be corresponding to the anticodon arm. This anticodon arm is the one which will recognize the codons in the mRNA. Okay. So this mRNA and this antibody will be in sync. They will be in sync. This mRNA codon and antibody will be in sync. So each tRNA which is charged will carry a particular amino acid. Okay. So what will the process of translation is, every single amino acid tRNA except the initiator one. The initiator, the first one. The first one will come and sit in the P site. First one will come and sit in the P site. Okay. Before, in initiation, first the 30S only will be there. Okay. After the initiation factor 3, there is an initiation factor 3 which will prevent the 50S from joining with the 30S. Okay. So there is 3 sites I told, no? P site, E site, E site, A site. Okay. So once the first amino acid is coded by a codon called as AUG. AUG is an initiator codon. This AUG codes for, in prokaryotes, it codes for an amino acid called as F-methionine. Formyl methionine. Most of you will be knowing that methionine is the first initiator amino acid, but the difference is, in prokaryotes it is formyl methionine, in eukaryotes it is verum methionine as such. This point was asked as a true or false statement in AIMS, I think in 2016 or 15, somewhere one question is there. So even such subtle details you have to remember. Okay, so this particular tRNA which carries F-methionine has to come and sit in the P side. This is the step. Until this happens, this 30S and 50S will not be allowed to join together. This is taken care of by an initiation factor called as IF3. Okay. Then there are some 2 factors called as IF2 and IF1, which will guide all the new incoming tRNAs to the A site. So once the setup is like this, the first codon is going to the first tRNA carrying the F-meth is going to the P site. New will come and sit in the new tRNA will come and sit in the A site. The initiation factors will disengage. They will disengage. So next will transport. Next will move towards the elongation segment. In elongation segment, what will happen is, supposedly, so here we have the methionine. Okay, first. Okay, A site, P site, sorry, E site, P site, A site. Now here, new amino acid has come to the A site. Okay, now what will happen is, this new amino acid containing tRNA will undergo a rotation to this direction. It will undergo rotation. Now this amino acid will come like this. For this, we need a GTP. What will happen is, here one enzyme is come and join these two together. This enzyme is called as peptidyl transferase. Now what will happen? This tRNA will become bald tRNA. And we have 2 peptides here. Now what next will happen is there is a translocation movement where this tRNA will give one push to this tRNA. Okay, so this tRNA will go from P site to E site, and this tRNA containing the 2 amino acids will be here, and new tRNA will come and join in the A site. So this movement is called as one push is called as translocation of the tRNA from P site to E site, and the vacancy of A site for the allowing incoming new tRNA to then same process. This will come rotation, then peptidyl transferase, then 3 peptides give a push, then A site will become. Okay, this process is called as elongation. So when subsequent entity has been reached with a lot of peptides, see supposedly your target is 8 or 7 peptides, you have reached everything. There is one factor called as release factor. There's one factor called as release factor. This release factor will come and put a nick, and this will go out. This protein will be going out. This is the process of translation. Now our antibiotics which we use mainly will keep targeting only these steps of translation. If you want to inhibit, then it will transfer, it will prevent any part of this mechanism, so that it will start working against them. Okay, so what are the 4 antibiotics they have given? Chloramphenicol, erythromycin, streptomycin, tetracyclines. So basically how they work is chloramphenicol inhibits the peptidyl transferase activity. So chloramphenicol inhibits peptidyl transferase. Now it acts on the elongation step. It acts on the elongation. Erythromycin prevents the translocation process. I have explained you. Okay, so it prevents the translocation, the push from P site to E site and A site to P site. That is prevented by erythromycin. Okay, streptomycin binds to the 30S subunit and causes misreading of the mRNA, so that codons are not properly read. Okay, so what will happen? Codons are not properly read. So that means it is also acting in the elongation process. The chain will not continue. So the termination, it acts as an artificial termination for the chain to peptides to keep elongating. Whereas tetracyclines, they will inhibit the incoming new tRNA. So the new tRNA is coming, no, that itself it will prevent. So it will not allow the new tRNA to come and join in the A site. Okay, so basically the mechanism of antibiotics are like this. You can also go back and read mechanism of antibiotics of puromycin and diphtheria toxin and ricin. All these are extra, but predominantly the questions will be focused only on chloramphenicol, streptomycin, tetracyclines and erythromycin. So we'll again see the question: identify the protein synthesis inhibitor that prevent the attachment of amino acid tRNA to the A site. So they are asking new amino acid tRNA is coming, and it is, see, this is P site, this is P site, all the, see, so much peptides it is having. Okay, and A site is vacant. So one tRNA, new tRNA want to come and sit in the A site, but it is not allowing. One antibiotic is there. Which antibiotic is there? The antibiotic is nothing but tetracyclines. Okay, tetracyclines. You all know the mechanisms. Okay, streptomycin, misreading of the mRNA. Erythromycin, you have the translocation, the push, no, from here to here it will push, no, that is not allowed by the erythromycin. And chloramphenicol, the bond, this bond, it is formed, no, that peptidyl transferase inhibitor. Okay, fine. Right. Identify the following technique depicted below, which is used where originally developed as a research tool to explore and manipulate genes, but now used to identify defective genes associated with disease and to correct genetic defects. Okay, it's very simple. Why I wanted to put this question is not because of this question. I wanted to tell you about something else. Okay, you have to read about something else. Gene editing. That technique is called as gene editing. Okay, we'll come to that. Okay, see here. There is one DNA. You are taking one piece of this DNA. How you will take? By using scissor. What scissor you will use? You will use a scissor called as molecular scissor. The molecular scissor is called as restriction endonuclease. Restriction endonuclease. Restriction endonuclease. Somebody asked me, sir, you are putting echo RI. What is the meaning of this echo RI? First, it is not echo RI. It is called as echo R1. The first two letters tell from which species it's coming. Escherichia coli. That is the echo. R is a subtype. One is a sequence in which that enzyme is discovered. From Escherichia coli itself, R strain itself, so many resistant endonuclease has happened. So we are giving the number, the sequence in which it was discovered. Okay. Now this molecular scissor or the restriction enzyme goes and cuts the DNA at particular sequences. For this, it may be GATC sequence. Wherever it is, it will cut that kind of sequence. Okay, so the kind of cutting is there. It will generate two types of ends. One is called as a sticky end, another one is called as a blunt end. Okay, this much is enough. Okay, then you have cut that particular DNA characteristic which you want. Supposedly you want to make a baby clone with some particular characteristics. You know one person, that person is having a good characteristic. I want that characteristic in this baby. You take that particular DNA, okay, you cut it using the molecular scissors. Now you want to put that inside the baby. How you will put that inside the baby? You will take that particular DNA and use some material called as a vector. Vector is a carrier of DNA. Okay, this vector comes in 5 sizes, 5 different sizes coming. Okay, what are those? Plasmids, smallest vector, bacteriophages. Okay, then cosmids, then bacterial artificial chromosome, yeast artificial chromosome. This is the smallest, this is the biggest. Based upon your size of insert, based upon what kind of size you want to transport, that particular vector will be chosen. So here we are taking, let us assume we are taking small piece of DNA. You put the DNA, you put the vector, there is an enzyme gum, which is the gum morph, we call it molecular biology, DNA ligase. It will add together. Now this you take and you put it inside the baby's genome. Okay, so baby DNA is there, this new DNA with vector is there, so it will mix together and the baby will start producing the new DNA along with the old DNA. That characteristic will come. This technology is called as the molecular biology technique, recombinant DNA technology. This is called as recombinant DNA. So our product which are we are getting is called as the clones. The product which we are getting is called as clones. Okay, so microarrays are for genetic expression, whether a particular RNA is transcribed or not. Southern blot, I've already told you, it is for DNA blotting techniques, hybridization techniques. DNA sequencing is just like you are elaborating a particular sequence of DNA. What all nucleotide is there? They take one notebook and write C followed by G followed by A followed by T. This is called as DNA sequencing, done at a very advanced level. Okay, so this particular technology is called as recombinant DNA technology. However, the question now that is being asked widely is called as CRISPR-Cas9 technology, which is a gene editing technology. Gene editing technology. Okay, supposedly you have a particular gene. Okay, recently, last 1 or 2 years back, there is a scientist in the China. This fellow, what he did is he told that I produced a baby which cannot never get to the AIDS at all. He told like that. What happened? They put him in jail. Okay, what he did is he used this CRISPR-Cas9 technology and he take the 2 genes, he cut the parts which are susceptible to get infected by the RNA virus. This is what he claims. Okay, so this is a gene cutting technology, gene editing technology. You can add editing. See, gene editing, there are 3 processes. One is you can insert, second is you can edit, third is you can replace or repair. All these 3 can be done. So he deleted some genes which will make a person susceptible to HIV virus. Clustered, clustered, regularly interspaced, short sequence, palindromic repeats. Cas9. Okay, so clustered, regularly interspaced, short palindromic sequence repeats, or short sequence palindromic repeats. This is what is known as CRISPR-Cas9 technology. So this you have to understand. Okay, let's move to question number 8. The incidence of sickle cell anemia among blacks is 1 in 400. I told you this question. Calculate the frequency of heterozygotes for sickle cell gene encoding beta S globin in this population. Hardy-Weinberg law. So Hardy-Weinberg law is totally for autosomal recessive technology. Totally for autosomal recessive technology. Okay, so autosomal recessive inheritance. Okay, what this Hardy-Weinberg law states is, supposedly, I'll never forget this, supposedly a particular characteristic has two genes, okay, two alleles. The expressive form of genes is alleles, okay, two alleles, A and small a and capital A, okay, and the frequency of expression of capital A is P, the frequency of expression of small a is Q. You'll remember this, okay. So this particular alleles come from mother and father. So let's draw something called as a Punnett square, okay. So we'll consider like this. So the possible four types of expression are AA, small a, small a. Okay. Now if I substitute the frequency with this, then A into A will become P square. Then this will become PQ. This will become QP. This will become Q square. Correct? So the total possibility of this expression is P square plus PQ plus QP plus Q square equal to 1, or P square plus 2PQ plus Q square equal to 1. 100%. 1. Okay. Or otherwise, P plus Q the whole square equal to 1. Correct? Or otherwise, P plus Q equal to 1. This is the Hardy-Weinberg law. Okay. So the sum of expression, the expression of a recessive allele and a dominant allele put together comes as 1. Simple. Okay. It comes as 1. Sir, but you told only autosomal recessive. Yes. Okay. Why autosomal? Why not sex-linked? In sex-linked, we cannot determine. This has come from X chromosome, Y chromosome. The distribution is not there. There are certain particular rules for Hardy-Weinberg law. Okay. See, the population must be a fixed population. Okay, there should not be any immigrants. Okay, now you are supposedly we are applying the Hardy-Weinberg law to the Chennai population. You should not say that, sir, some people yesterday only coming from the America, they have started mating with the Chennai population. No, then this law will go obsolete. In Chennai population itself, random mating must happen. It should not be like consanguinity, sir, all our relatives only mating has happened. Sir, one particular group in Chennai itself, sir, they live alone. Within them, mating is happening. Let us compare them with all together. No, that is not allowed. So all these conditions are there. You should not, immigration should not be there. Mating should not happen within consanguineous group. Random mating only must happen. Then mutation should not be there. Okay, some particular mutation already exists in one group, that group is joining with this group. No, that is not possible. Mutation, the population should be freed of mutation. There should not be selection bias. So always autosomal dominant alleles have negative selection bias and recessive alleles have the positive selection bias. Nature tends to favor us by negatively selecting the dominant alleles. Okay, so because if the dominant alleles are there, then the inheritance will be very strong. Every generation will start getting the disease. So nature itself says, okay, let's not be so harsh on the humans. Let us only assume that it is not going to affect as severely every generation. Let's probably the disease person, let them stay small. Okay, so negative selection bias towards dominant alleles, positive selection bias towards recessive alleles. No consanguineous marriage, no immigration, no mutation. Select group of people and random mating must happen. Then only we can apply the Hardy-Weinberg law. Okay, now what he is telling is sickle cell anemia among blacks, homogeneous population. Okay, no white, only black. Okay, is 1 in 400. We should not be racist, but then what to do? Okay, 1 in 400. Calculate the frequency of heterozygotes for sickle cell gene. So I told you this law is applicable only to the recessive. So the recessive, this is dominant, right? This is heterozygotes. This is the recessive people. So this is a recessive disorder. So what he is saying is Q square is 400. That is what he said. So 1 by 400 is telling now. Then 1 by Q square is equal to 1 by 400. What we will do? We will do 1 by Q is square root of 1 by 400 or 1 by 20. All right. So this 1 in. Okay, we'll take it as 1 in. Okay, we'll take it as 1 in. Okay, so 1 in Q. How much is Q value? 1 by 20. Nothing but 0.05. Am I right? 0.05. Right. Okay, so Q value comes around, Q square is 1 by 400. Yeah, root of 1. Q square is 1 by 400. The root of 1 by 400 is 0.05. Now you all know the formula. P plus Q is equal to 1. So P plus 0.05 is equal to 1. P is equal to 1 minus 0.05, which is 0.95. So P square dominant, homozygous. Q square recessive, homozygous. Which is the only heterozygous thing? PQ and QP. So in this expression, go and submit. If you submit, this is the heterozygous population. So they are asking, what is the frequency of heterozygotes? Frequency of heterozygotes is PQ plus QP, correct? Or 2PQ. So what we found out from the question, we found out that 2 into P value is 0.95, Q value is 0.05. Okay, how much is that? Let me calculate. 2 into 0.95 into 0.05. It is 0.095 or roughly, okay, 0.01. I can keep it as 0.01 or 1 by 10. Correct? 1 by 10, whatever it is. 0.1 or 0.095, no, rounded na 0.1. So it is 1 by 10, which option is very close to that? 1 in 6, 1 in 11, 1 in 20, 1 in 22. If you divide exactly, means it will come as 1 by 11 only. So I'm telling you roughly, very roughly, it is 1 in 11. So this is how you have to solve this question. Okay, so go back and read. This is all classic USMLE questions. From this year onwards, I'm expecting these questions to surface in Indian exams also. Okay. All right, very nice. Based on the carrier given data, which is the most likely mode of inheritance for this disorder? Autosomal recessive, autosomal dominant, X-linked recessive, X-linked dominant, mitochondrial. 5 options are there. Okay, let me give you 6 golden rules for pedigree chart. Pedigree chart, how to decode a pedigree chart. Point number 1. So Balas rules. Okay, Balas rules, biochemistry rules. Good. Point number 1. If the affected males equal to females, then it is autosomal disorder. Okay. If the males are greatly affected or females are greatly affected, then it is a sex-linked disorder. This is rule number 1. So first, you all know what is male and what is female. No, round is female, square is male. Okay. Minimum 3 generation data is necessary for a pedigree chart. Usually you will get 3 or 4. Okay, that they will take care. You don't have to worry. But if you are becoming a question setter, at least you should give minimum 3 generations of data. Okay. So male and female equally affected, autosomal. Male and female, only males affected or only females affected, it's a 6-linked. Okay. Number 2. If the disease is present in all generations, every generation is affected, then it is a dominant disorder. If the disease skips generations, one generation fully there is no disease, then it is recessive disorder. It is recessive disorder. Number 3. If only father transmits the disease to the offsprings, that too father transmits only to the son, then it is Y-linked. If only the mother transmits to son or daughter, then it is mitochondrial. Then it is mitochondrial. Okay. Number 5. Carriers are considered as normal persons. They are not diseased, they are considered as normal persons when it comes to our rules. Okay. Number 6. If a deceased child is born to normal parents, child deceased, parents normal, then this is recessive disorder. Then this is recessive. These are the 6 points you have to remember. Okay. Point number 1. Males and females equally affected, the autosomal. Males and only males affected, only females affected. Think about sex linked. Okay. Number 2, all the generations have the disease, then dominant. One full generation, there is no disease. It is recessive. Number 3, only the father is transmitting the disease. Think about Y-linked. Father transmits to who? Son. Father transmits to son. Father is transmitting to daughters also, then you should go and think about X, but don't worry about that. Father transmitting son means it is Y-linked. Mother is transmitting the disease. Father is never transmitting the disease. Mother is disease, then the disease going to the son or daughter, both, any of them, then becomes a mitochondrial. Okay, carriers are always considered as normal person. They are not disease person. Carrier will be denoted like this or like this. Always look out for carriers. It's very important. Okay, child is diseased, but the parents are normal, means it is recessive disorder. With this backdrop, if you go on immediately, you will understand what to do with this. Look at this. Look at what has happened here. Okay, we have a carrier here. 4 generation data is given. Carrier here. See, every generation has a disease. Every generation has a disease. Now, who's transmitting a disease? Let us see. Let us take this child. Let us take this child. Let us take this child. Okay. By looking at their parents itself, I can tell even this child. See, this child parents, both of them are normal, but the child is diseased. This child parent, both of them are normal, child is diseased. This child parent, one is carrier, one is normal. That's okay, but carrier is considered as a normal person. So both the parents are normal, child is diseased. Rule number 6 says when both the parents are normal and the child is diseased, it is recessive disease. But then look at the chart. Only males are affected. Oh, only males are affected. Okay, so which means it is a sex-linked disorder. It is not autosomal disorder. It is sex-linked disorder. So this is the answer is X-linked recessive. Now some of you might come and ask, sir, you told every generation must be affected. I mean, if a disease, every generation is affected means it is dominant. This generation is affected, this generation is affected. But what about this generation? This generation is never affected. That is why I am saying it is not dominant. It's a recessive disorder. It is a recessive disorder because it has skipped this generation. It is a recessive disorder because the child is born to normal parents. It is a recessive disorder that is because, it is a sex-linked disorder because it is affecting only the males, not females are affected. It is a sex-linked recessive disorder. The only option is X-linked recessive. So the answer here is option C, which is X-linked recessive disorder. Mothers are not at all affected, so it is not mitochondrial. Okay. So you all understood this. Very nice. A 5-year-old boy has a rough raised lesion on his neck, has excessive freckling and some erythema on his face, lips, neck and upper extremities, as well as some clouding of his corneas. His mother reports that he has a tendency to get sunburn easily. Pathologic evaluation of a biopsy reveals it has to be a malignant melanoma. In this case, which repair mechanism is most likely defective? Base excision repair, mismatch repair, nucleotide excision repair, direct repair. Okay, so it's about a DNA repair mechanisms. In DNA repair mechanisms, very simply classification: single strand repair, double strand repair. Usually I teach like this: single strand repair and double strand repair. In single strand repair, 4 categories are there. In double strand repair, 2 categories. Single strand repair, category number 1, mismatch repair. Category number 2, base excision repair. Category number 3, nucleotide excision repair. Category number 4, direct repair. Okay, mismatch repair. See, the basic problem in DNA repair is, supposedly there is an error occurred, somebody will come, cleave that particular portion of the DNA or whatever the material, they will replace it with new, they will seal it with DNA ligase. Okay, the problem in mismatch repair is a particular portion of repair, I mean this one is not matching. G should be paired with C, it will be A, something like that. So because of this, there will be a distortion. Okay, and some enzyme will come and endonuclease will come and it will cut that particular portion. Then DNA polymerase will come and replace the correct nucleotide, and then it will seal with ligase. This is mismatch repair. Base excision repair, cytosine single base. It's only single base. There it is a sequence. Here it is single base. The cytosine spontaneously deaminates to uracil. Spontaneously deaminates to uracil. Okay, this uracil is not sustainable in a DNA molecule. I said thymine only stays in the DNA, uracil stays in the RNA. So there is one nucleus, endonuclease, which comes and removes the uracil remover. That is a uracil remover. Okay, glycolase, uracil glycolase inhibitor something. So it will come and this will leave a gap. This gap is called as a AP site. AP site. Okay, apurinic, apyrimidic sites are called as AP site. Then DNA polymerase will come and fill that one. That then it will seal with the ligase. In nucleotide excision repair, the pathophysiology is nearby two thymine residues are there. These two will form a dimer instead of binding with A. So T should bind with A, no. Instead of binding with the complementary nucleotide, these two will form a thymine dimer effect. Dimer adduct. So this will happen when the DNA is exposed to UV rays. This will happen when the DNA is exposed to UV rays. Okay. In direct repair, sometimes there is guanine residue which is methylated. So there is an enzyme which will come as methylguanine methyl transferase. This enzyme is called as MGMT. So when a nucleotide gets methylated, its expression will come down. It cannot express itself. Thus methylation is an inhibitor. Like, okay, so this methylated guanine will be converted into normal guanine, then it will go. So these are the 4 single strand repair mechanisms. Okay, these are the single strand mechanisms. In base excision repair, you have the HNPCC. Hereditary neopause non-coli. Then in double strand repair, there is only 2 things: homologous end joining, non-homologous end joining. So supposedly the strand is like this, both of them repaired end to end. That is homologous end joining. Non-homologous means strand is there, then it will cross over and repair like this. This end is called as holiday junction. Okay, this is called as non-homologous end rejoining. I'm not going to much details about that. So this particular person, he is suffering from disease, freckling, erythema, upper extremities, and some clouding of cornea, sunburn. All these point towards only one thing. This disease is, xeroderma pigmentosum. Classic defect seen in nucleotide excision repair. So that exposure to sunlight. So I told you it's exposure to UV rays. Okay, xeroderma pigmentosum, ataxia, telangiectasia, all these are diseases coming under this category. So if that particular mechanism of nucleotide excision repair is defective, then it lands with diseases like this. It's a very classic picture. Looks like malignant melanoma, but then freckles will be there. Sun exposure is not tolerated well. All these things are classic features of nucleotide excision repair mechanism defect. The disease is called as xeroderma pigmentosum and then ataxia. Sometimes you may get a defect in DNA ligase or this cross joining also, Bloom syndrome, Fanconi's anemia. All these things are also coming under the DNA repair mechanism defects. Okay, so I hope this session has given you enough insights into what is the basic pathophysiology and the basic molecular biology mechanisms and techniques. Now, let me conclude this session by saying that molecular biology questions are not very beyond you. They are within your reach, only that you need to understand the concepts, you need to apply yourself, and you need to have a clear thought of mind. It's very difficult to crack a molecular biology question with a clouded mind. It is very difficult to crack it without knowing concepts. But if you know the concepts and if you're cool, then I don't think molecular biology is tough at all. It can be made more easy. It can be made more feasible to learn also. Hope to see you soon in one of our classes. And until then, stay safe, stay good, and bye. Thank you.